right, am I at the right screen?
We can see your notes on. There we go. There we go. That's it.
All right, awesome. So thank you, Matthew, again for putting this series together. I think this is a wonderful platform, and I am really appreciative of the opportunity to share with you all the object of my affection, the study of interferon-mediated inflammation. And today I'm going to tell you a bit about how we approach basically the study of interferons. So to start out, canonically or classically, we know interferons to be potent antiviral cytokines that are induced by nucleic acid recognition that's stemming from either viral infection of the cell, whether this be RNA, DNA that's sensed by pattern recognition receptors, or the sense of self-DNA that stems from dying cells. However, we now can understand as well that there's other stress responses that can lead to the induction of interferons through the activation of transcription factors such as the IRF family of transcription factors and NF-κB. So I've spent most of my training really trying to understand how, whether a viral infection is in its chronic stage or whether it's an acute infection, how that shapes the outcome of the interferon response, whether host or pathogen genetics affect how the interferon response is mounted, but also going back to this idea that not only do we have pathogen-specific responses, but also stress-induced interferon responses, how these can control the induction and response to interferons. And as I was mentioning, we generally think of interferons as friends because they do promote the clearance of pathogens and therefore actually protect the host with generally very minimal cost of fitness. However, when interferons do rear their ugly side, what we can see is that at times it can actually promote viral persistence. This is something that was shown in chronic infections, as well as actually cause pathogen and inflammation associated sequelae that could have long-lasting effects to the host. So why does this actually happen? So we think of interferons actually as being the salt of the earth because they do have pleiotropic effects in the regulation of immunology. And as we've been presenting thus far, yes, they do regulate the expression of interferon-stimulated genes, which are effector genes tasked with blocking viral replication and dissemination, but they've also shown to be protective against bacteria and fungal pathogens. Interferons actually start to bridge the innate to adaptive immune arms by regulating the expression of chemokines that will recruit cells into sites of infection and activate them in order to promote antigen presentation and pathogen clearance and establishment of memory. But most importantly, also, interferons can regulate cell proliferation and apoptotic responses. Therefore, they can promote repair at barrier sites after infection, but they can also be and have been used as anti-proliferative cytokines because of this powerful effect. And so when interferons become dysregulated, what we start seeing is actually quite the opposite effect, is that they can actually fuel in certain contexts the pathogenesis caused by certain microbes. The dysregulation of interferon responses can lead to hypercytokinemias, and this is a lot of what we see in autoinflammatory and autoimmune diseases, especially the autoinflammatory diseases called interferonopathies because of the induction of interferons. But they can also be immunosuppressive, causing immune cell energy. And by virtue of being able to disrupt cell proliferation and differentiation, we can actually impair tissue damage. They can actually impair tissue repair and promote more damage. And so really, this has been one of the key questions that my lab tries to address is what really determines that balance of interferon between being protective versus being pathogenic. And this brings us to really going back and understanding that when we talk about interferons, specifically cell intrinsic and antiviral interferons, we're talking about two different families of cytokines. And so we really need to start understanding now what is the specification of function of both the type 1 and the type 3 interferons. These are cytokines that signal through distinct receptors, the type 1 interferons signaling through a heterodimeric receptor composed of IFNR1 and IFNR2, and the type 3 interferons or interferon lambdas, which are part of the IL-10 family of cytokines and signal through the interferon lambda receptor 1 and the common chain IL-10R2. But what they both have in common, these two receptors, is that they use the JAK-STAT signaling pathway to promote the activation of the transcriptional complex ISGF3 that's composed of STAT1, STAT2, and IRF9 to drive the expression of these effector genes, the interferon-stimulated genes. And so for a very long period of time, it was thought that these two cytokine families basically had redundant effects. Until most recently, we've started to appreciate that while the type 1 interferon receptor is ubiquitously expressed in most nucleated cells, the interferon lambda receptor high affinity subchain IFNLR1 is actually tissue restricted. It's the highest in expression in epithelial cells and human hepatocytes, but we've come to appreciate that in immune cells, its expression seems to be abundant enough to confer responsiveness to type 3 interferons, and if anything, could also be induced. But more importantly, work from Evangelios Andriakos' lab and Andreas Wack, they've shown that there's actual subtleties and unique functions that are exerted by both the type 1 and type 3 interferons. So in a really elegant study, Eliona Galani actually showed that upon very low levels of infection, the predominant interferon species that are induced are the type 3 interferons or interferon lambda. And this can actually clear viral infections with limited inflammation. However, upon viral escape and increases in viral burden, what happens is the type 3 interferons are no longer capable of preventing viral dissemination. And now we have the induction of type 1 interferons that can cause very inflammatory responses that actually are costly to the host. And so the project started in keeping this in mind and thinking about the evolutionary requirement of both type 1 and type 3 interferons, and this discrepancy in their ability to induce inflammation, asking a very specific question then. What distinguishes their ability to mount this inflammatory response? So first we started asking a very simple question. We know that the type 1 and type 3 interferons actually display kinetic differences in their regulation of ISG. So we thought maybe this is all there is to it. And so at first hand, what we looked at was the ability of the ISGF3 complex to bind to ISRE elements using an electromobility shift assay. And so here we use the ISG15 ISRE sequence as a probe to try to interrogate whether type 1 interferons, as we'll be using interferon beta in this case as a putative type 1 interferon and interferon lambda 3 as a type 3 interferon, and ask, do we get complex formation? And we saw that indeed in treatment with either beta or lambda, we saw as early as 3 hours formation of the ISGF3 complex that was capable to bind our probe. However, by 20 hours of stimulation, we saw a resolution of these complexes that was still sustained in response to interferon lambda treatment. Intriguingly though, we saw that while both cytokines were capable of this early induction of ISGF3, type 1 interferons were much more robust in actually inducing ISG protein accumulation, where we had higher expression of ISG15 and much faster accumulation of ISG15. And at later time points, we started to see that resolution that we see by 20 hours, where protein expression starts to dissipate, while type 3 interferons still continue to maintain the ability to both transcribe genes as well as promote their translation. And so broadly, we think then that at barrier sites, we need the collaborative effect of both type 1 and type 3 interferons because we have a very quick burst of ISG expression in response to type 1 interferon or interferon beta, as we can see here in this antiviral protection assay. So here we have pre-treated cells for the allotted time, so the indicated time, prior to infection with a cytolytic virus, and then we're examining how many cells actually are protected from lysis stemming from the virus infection. And we see that with interferon responses as early as 6 hours with type 1 interferon, we get robust antiviral protection, and these weans as we see weaning of ISG expression, and this is expected as we know that the type 1 interferon receptor actually undergoes ligand-mediated downregulation. However, with type 3 interferons, we did not see a robust protection by 6 hours. We actually started seeing that it took about 24 to 48 hours of pre-incubation with type 3 interferons to attain protective levels, and together in the combination, the presence of both, what we see now is that we can maintain an antiviral threshold. But it still doesn't explain then why are they, if type 3 interferons can sustain the activation of ISGs, why do we still not see inflammation? And what we started seeing then is that there was a set of genes that were biased towards responding to type 1 interferon stimulation, but they did not seem to be robustly induced by type 3 interferons. And a subset of these genes were actually the CXCR3 family of ligands that is responsible for recruiting monocytes and T cells into sites of infection or damage. And these were CXCL9, 10, and 11. And we saw that we had a very robust response of these genes to stimulation with interferon beta, but we did not see a very significant induction by type 3 interferons, nor did we see a delay as we saw in other genes like MX1 or ISG15, which are also ISGs that respond to both cytokines. And so this brought me to my expertise, transcriptional regulation, and understanding how the interferon responsive factors, or the IRFs, actually regulate ISG expression. And what we found was that the transcription factor IRF1 seemed to be predominantly induced by stimulation with interferon beta, but it was largely unresponsive to interferon lambda in immortalized human hepatocytes. We looked at the expression of other IRFs that are involved in the induction of interferon-stimulated genes downstream of interferon receptors, like IRF9, which is part of the ISGF3 complex and is also in itself an ISG. And we could see that both interferon beta and interferon lambda could actually promote the accumulation of IRF9. We looked at a second ISG as well, but also regulator of positive regulator of ISG expression, IRF7. And again, while we saw that its expression was maximal in cells that were stimulated with interferon beta, it could be induced by stimulation with interferon lambda, and much like other ISGs, which was also maintained over a large period of time. So we then asked, is this just a property of interferon beta and interferon lambda 3? And no, that's not the case. We can generalize this bias of IRF1 responsiveness to type 1 interferons being very potent inducers, whereas type 3 interferons are very weak inducers, if anything, of this transcription factor. More importantly, because we were looking at human hepatocytes, which in this case are express the type 3 interferon receptors, unlike mouse hepatocytes, we also wanted to know, this is something that could be species conserved. This is also conserved across other barrier sites. So we also used various models of intestinal and airway epithelial cells, both primary cells and cell lines, to show that we did indeed see a conservation of this bias across different barrier sites. So is IRF1 actually essential? And the reason for this pro-inflammatory response? so we now used CRISPR-based genetic knockouts of IRF1 on our hepatocyte lines. And what we saw was that IRF1, while being important for early antiviral protection and for the earliest burst of ISG inductions that is promoted by type 1 interferon sensing, it was actually not necessary for the later stages for prolonged antiviral response. And this explains why, in the absence of IRF1, we're still seeing a robust enough antiviral response that is protective stemming from type 3 interferon regulation. But what we did observe by doing genome-wide transcriptomics was that, whereas we again see that the induction of chemokines and cytokines and other genes that are through a gene set enrichment analysis shown to be important for both the recruitment and activation of immune cells, while these responses seem to be biased towards type 1 interferon and absent in interferon lambda, we absolutely, and we could see this in vivo, actually, with stimulation of intranasal inoculation of mice in lungs. We could see that induction of interferon responses by inoculation with interferon beta was sufficient for the recruitment of monocytes, macrophages, and dendritic cells, which was largely not seen in response to interferon lambda. These responses were definitely IRF1 dependent, and we could see these that both the expression of CXCL10 was largely dependent on the ability of cells to enhance IRF1 expression, as well as that of transcription factors like C2TA that are important for antigen presentation. So what determines these discrepant response? So we really wanted to get at, okay, is there something that is more proximal to the interferon receptor? And we went back to asking, are there differences in the regulation or the activation of STAT transcription factors? And so when we had started the comparison between the two responses, one of the things that we wanted to make sure was that we had the same amount of proximal activation of the transcription factor STAT1, and despite that, we were still seeing such a difference in interferon stimulated gene expression. So we wanted to then get at whether, we were seeing different transcriptional complexes being formed in response to either type 1 or type 3 interferons. So we had started our study again looking at ISGF3, and we had demonstrated that both of them were able to induce, both cytokine families were able to induce a robust transcriptional complex. But then we really were very intrigued by this bias and the potential that perhaps type 1 interferons were exquisitely capable of inducing STAT1 homodimers that are associated with driving the expression of genes that have Gas elements in their promoter regions rather than ISRE elements in their promoter regions. And this we know too, that that's how interferon gamma can actually be such a robust inducer of both CXCL10 and IRF9. But we were also curious to see whether perhaps what was happening is that other transcription factor complexes, such as STAT1 and STAT3, could be actually formed by interferon lambda preferentially that were actually blocking this ability to induce CXCL10 and IRF1. So again, we used an electromobility shift assay, or EMSA, to interrogate this question. And now we're using the IRF1 gas element. And we see that, yes, in response to our positive control interferon gamma, we see binding and super shifting of our DNA protein complex when we incubate with antibodies against STAT1, but we did not see any changes when we incubated with any other antibodies against other STAT proteins. And again, this was similar to, we could observe the interactions of a STAT1 homodimer that was shiftable again with STAT1 antibodies. And this complex was absent in response to interferon lambda, and we did not see any evidence of any complexes that could be STAT1 or STAT3 dependent. We also showed that interferon lambda co-incubation with interferon beta does not actually inhibit the induction of IRF1. So there was not a reason to believe that perhaps there was an active inhibitory signal downstream of the interferon lambda receptor that prevented IRF1 induction. So what sets this threshold of inflammation? We wanted to ask whether the limiting factor then was the abundance of the high affinity subunit IFNLR1. And here what we did was look at IRF1 induction in cells that are competent still to respond to both type 1 and type 3 interferons because they express normal amounts of interferon lambda or basal amounts of interferon lambda receptor 1. And upon IFNLR1 exogenous expression, what we see is that the cells become much more responsive to type 3 interferons, as you can see here. The activation of STAT1 actually becomes much more robust. And now we start seeing induction of IRF1. Again, supporting this idea that there is not necessarily an active repressive signal, but simply there is not enough type 3 interferon receptor available to actually lead to both the induction of the ISGF3 complex and abundant STAT1 activation to promote homodimer formation. And of course, this overexpression of the lambda receptor actually increased both the responsiveness to canonical ISGs that are very responsive to interferon lambda stimulation, but also greatly enhanced the expression of CXCL10 after interferon lambda 3 stimulation. Interestingly, though, what we did not observe in vivo in influenza infected murine lungs was the ability of viruses to induce the expression of interferon lambda receptor. And this was in stark contrast from work from Dr. Emily Heeman's lab, where she shows actually that in immune cells, in dendritic cells, influenza infection is actually capable of inducing the expression of the lambda receptor. We try to ask then whether this could be happening in vitro, and again, we did not see any responsiveness, positive responsiveness of the interferon lambda receptor 1 subunit in response to pro-inflammatory stimuli such as TNF-alpha, poly-IC treatment, or Sendai virus infection in our immortalized cells. And again, this is in contrast with work from Diana Santer's lab, work from Golo Ansteel's lab, where they've shown that actually ex vivo or in vitro stimulation of immune cells can actually induce interferon lambda receptor. And so this leaves a very open question in the field of what actually determines this discrepancy between the epithelial barrier, where you want to maintain that threshold of inflammation, versus that of immune cells, where you might want to tap into these interferon lambda responses in order to enhance protection. And so with that, I hope that I've been able to convince you that IRF1 in the context of interferon treatment is actually the master regulator of inflammation. Its induction is biased. When we think of antiviral responses, is actually biased towards the type 1 interferons that can tap in a program that's similar to that of interferon gamma or the type 2 interferons, where the induction of the STAT1 homodimer actually induces the expression of IRF1. And this is why both type 1 and type 2 interferons are so exquisitely inflammatory, whereas type 3 interferons, by virtue of low levels of interferon lambda receptor 1, don't necessarily surpass that threshold, and so they retain antiviral activity and the same antiproliferative activities that are attributed to all interferons, but with a very limited or muted inflammatory response. But I would be doing you all of this service if I didn't tell you then, if we're looking at the specific external function of these interferons, why then do we need interferon lambda beyond just not surpassing inflammation? And that's actually what we found was very interesting is that yes, there was indeed a subset of genes that seemed to be sustained or more biased towards the type 3 interferon response that seems to be skewed towards the control of cell proliferation and differentiation. We, by doing upstream regulatory analyses of our transcriptional profiles, we've identified a subset of transcription factors that could be regulated preferentially in response to interferon lambda that are not thought to be canonical regulators of the lambda response, that seem to also be correlated in conjunction with other transcripts that seem to be regulated by MAP kinase signaling. So what my lab is doing currently, or what my lab is focused on, is trying to identify these non-canonical responses and how MAP kinases specifically can either enhance or inhibit the antiviral activities of the antiviral canonical activities of the type 3 interferons, but also how they can promote the integrity of the barrier, specifically focusing on what could be some of these transcription factors that could be co-regulated by the MAP kinases. And something that's really important to us as well in the barrier, where we have cells that are of cellular identities, mixed cellular identities, we now know and have shown very recently, we were lucky to collaborate or be part of a study led by Jeff Hill at the Fred Hutch, where they were looking at specific responses between stem cells, intestinal stem cells versus fully differentiated enterocytes, and looking at whether interferon lambda actually could elicit distinct transcriptional profiles, and indeed that is the case, as determined by transcriptional profiles derived from LGR5 positive versus LGR5 negative cells. So I hope with this I can leave you with really thinking about cell identity as a determinant to cytokine signaling, right? We've discussed that there's some cells that could be more predominantly biased to towards type 1 interferon responses, mucosal cells where you have interferon lambda responses in certain contexts being the primary regulators of inflammation, or cells where you can actually induce the expression of the type 3 interferon receptors. And how this would greatly determine essentially the robustness with which these core transcription factors that regulate the antiviral response are elicited, how inducible transcription factors that can enhance and that can amplify these responses, such as IRF1, IRF7, how this can be co-regulated differentially when we think about cell identity. But more importantly, what we're really after now is, what are those cell-specific states and cell-specific transcription factors and expression of core regulatory receptors? How can they actually modulate the response to interferon and really tip that balance between interferons as protective cytokines versus interferons as deleterious tissue damaging signals? And with that, I would like to end and take any questions, please. If you have any comments or insights or questions that remain after the discussion, please don't hesitate to email me. Big thanks to all collaborators and our sources of funding.
Thanks so much for that. That was a really, really interesting talk. Really enjoyed that. So we've already got a couple of questions. So one of them was just on your acknowledgement slide, Jacob. So he's asking, have you or anyone else infected IRF1 knockout mice with any viruses? And if not, do you predict the mice would fare better or worse?
So I have not done this myself, thank you, Jacob, but others have. They actually do fare worse. So from our data, what we do see is that the basal expression of a certain subset of ISGs is actually set by IRF1 expression. And so when you do remove IRF1, your overall levels of interferons actually are much lower. IRF1 is also necessary for proper immune cell development. So there's a key subsets of lymphocytes that are actually not well developed in the absence of IRF1. So broadly speaking, they fare worse.
That's really interesting. So we've also got a question from Fawaz. So do you want to go ahead and ask a question?
Sure, thank you. Thank you very much for the great talk, Adriana. My question was about the kinetic that you showed for formation of the complex. And I wondered if you knew how long it took for the complex to break apart.
No, so we did not do intervals shorter than that, and that's definitely a really intriguing question, because if you look bulk at the activation of stats, you can see that about at 4 hours you see lowering levels of stat 1 and stat 2 phosphorylation, and they get sustained actually in response to type 3 interferon. So the question is, when we start seeing those decreasing levels, is that when we're actually seeing later on? So does this take 5 hours? By 12 hours, actually you start seeing all ISG starting to decline. But we didn't really chase it with either chromatin immunoprecipitation assays or MSAs to really define that the complex was inactive.
And can I squeeze on just one more question? Do you know if there's any non-infectious pathogens that lead to the same, not non-infectious pathogens, non-infectious stimuli that treat to the same complex formation and dynamics? So things in like aging or autoimmunity?
Yeah, so actually, DNA damage response, we've shown in the past that DNA damage responses can induce type 1 and type 3 interferons. But they can actually induce, independently of interferons as well, the activation of IRF1. So IRF1 is a very dynamic transcription factor, because it is both downstream and upstream of type 1 interferons. And so it's hard to uncouple it from, what came first, the induction of interferon or the type 1 interferon or the induction of IRF1. But also in the cancer field, we're actually seeing that the induction of IRF1 can actually be counterproductive, because one of the IRF1 target genes is PD-L1. So while we want to enhance interferon responses, it could be best to try to enhance type 3 interferon responses in tumors to prevent the activation of IRF1.
Thanks so much for that. And yeah, thank you again for a great talk. If anyone has any questions for Adriana, then you can, I think you're going to put your, oh my God, I can't speak. Put your contact details in the chat box. And with that, we are going to move on to the next talk of this session, which is by Ed Roberts. And Ed is based at the Beatson Institute for Cancer Research and is going to talk to us about dendritic cells in anti-tumor immunity. So I'm going to hand over to you, Ed.
Thank you. Let's see if this is okay. Is that the right screen?
It is, yeah.
Okay. Well, thank you, Matthew, for organizing this. This has been really interesting. And it's nice to see so many people, different people, after spending so long in my flat. So what I want to do today is talk to you about some of our work on how dendritic cells cooperate with each other in driving effective anti-tumor immunity. And when I say how they do it, what I really mean is how they don't do it, and why we have tumors growing out even though we have T-cell responses against them. So I've become very used to speaking to cancer people. So if I'm teaching you all to suck eggs, which is a very British saying, if I'm telling you stuff you already know, feel free to jump into the Q&A and I'll shoot ahead. But just to give you the background, obviously dendritic cells have a key role in bridging between sort of the innate and the adaptive immune response. You have these cells that exist in the tissue. They constantly sample and have a lot of pattern recognition receptors. When they find something that looks bad, They mature, they upregulate co-stimulation, they move to the lymph node, they find naive T cells guided with the FRC network. They prime those T cells, help them differentiate correctly. Those traffic back to the tissue, and there the DCs help maintain their function. Now, before I carry on, I'll just point out that when I'm talking about dendritic cells, what I'm really meaning is CDCs, or conventional dendritic cells. So a lot of work has been done on monocyte-derived dendritic cells and also plasmacytoid dendritic cells, which are obviously very relevant, especially to our previous talk. But with more understanding of lineage and sort of functional differentiation, these probably don't perform this kind of central role that we think of for dendritic cells. You know, plasmacytoid DCs are very good at producing tons of type 1 interferon, possibly others, I'm not entirely sure. Monocyte-derived DCs are driven in by inflammation and have some antigen-presenting functions, but really, if you were talking about the cells that are involved in taking the antigen to the lymph node and driving the initiation of responses, it's really these conventional dendritic cells. And conventional dendritic cells sort of fall into two main buckets. You've got conventional DCs1 and CDC2s. And the kind of, almost the dogma is that these CDC1s are really specialized for cross-presentation of antigen. They're the ones that are going to be really good at driving CD8 T cell responses. And actually looking in tumors, when we put in tumors that express OVA, and then we take out these different DCs. So just for context, the CD103 DCs are migratory CDC1s, and the CD8s are lymph node resident. And the CD11Bs are various CDC2s. What we found is that when we label up these OT1s, put them with the CDCs, it was really migratory CDC1s that cause upregulation of NR77 and then division. This kind of fit the established dogma, and we showed that these were the cells that were critical for the initiation of anti-tumor CD8 T cell responses, which fit the general trend of what people thought CDC1s are doing. We then later had another paper where we looked at the CDC2s and found that these really were the best at priming CD4 T cell responses. And again, this all fits with the kind of general ideas that people had about CDC1s and CDC2s. The thing that was that's becoming more obvious though now through just a ton of different work is that this dichotomy of function is really not very, isn't a satisfactory way of looking at dendritic cells. So I've included here just an example of this from a review that Yanni Borst did along with Kastenmuller a few years back. And what they were sort of pointing out is that as your immune response develops, we're moving from left to right, you start with these interactions with migratory DCs that lead to some initial priming. They then activate lymph node resident DCs, and these can do things like coordinate T cell help. So these can be licensed by the CD4s, and those then drive effective antiviral responses, as they showed in that paper. But other people have shown there are other cells also involved in this kind of coordination, I mean, including plasmacytoid DCs, which again, harkens back to what Adriana was saying, these being important, interferons being really important for antigen presentation, where plasmacytoid DCs have been shown to help orchestrate these interactions too. But the work I'm going to present today is really the what I did towards the end of my postdoc in a collaborative project with Megan Ruland, who's now started her own lab at OHSU. And what we were really interested in was this sort of one step in the middle of this graph from Yanni Borst, which was looking at the idea that migratory DCs pass antigen to lymph node resident DCs. And the reason we were interested in this is that we're not really sure how lymph node resident DCs become activated. We're not really sure what role they play in driving effective immune responses. And we were thinking that if we could work out how they received the antigen, we might understand some of the other things about what other information could be encoded along with that antigen. So how could a cell that has come from the site of infection and has all that information in it, how does it transfer that information along with the antigen to these DCs that have never seen that infection, never been near there? And then how does that impact what's going on in tumors? And this is particularly relevant for us because we've looked at how T-cell responses to tumors are generated. And even though, as I showed in that previous slide, we can see that they do drive a proliferative response from the T-cells, what we don't get is a really good sort of, we don't get robust cytokine production. We get sort of a less than optimal T-cell response. And so what we want to understand is how these DCs are interacting specifically in a tumor setting to see if there are any sort of nodes that we might be able to poke at to try and improve that outcome, which could then synergize with other approaches that maybe look in the tumor. So to give you some background, I'm sorry I couldn't find a nice diagram for this, so you're stuck with a whole thing of text. I sort of went through the literature and looked at what were the different possibilities that people had looked at before. And so people have suggested that either we might get direct drainage of antigen, the migratory DCs come in, apoptose, and then resident DCs pick up the kind of cellular corpses. Others had suggested that monocytes brought the antigen in, and then there was sort of two similar possibilities that DCs came in and just chucked out exosomes or chucked out soluble antigen. And then there was the possibility of cross-dressing. For anyone who doesn't know, I do not recommend Googling cross-dressing DC and expecting to find out about dendritic cells. What you will find is, like I did, where to buy very large high-heeled shoes in the US capital. But cross-dressing is obviously where DCs come in and transfer fully loaded MHC molecules from them to the resident DCs. So these had all been suggested, and we thought they all had fairly different, they would all have fairly different consequences for how antigen is transferred and what information came with it. So this is what we really wanted to look into. So to do this, we generated a tumor cell line that expressed an incredibly bright and very stable fluorescent protein called ZS green. This is just an example. We put this tumor into mice that were MTMG mice. So these express a myristylated TD tomato. So every membrane of a host cell is red. So we let the tumor grow. You could see these big green swathes of tumor cells, and where they came into contact with sort of host immune cells and things like that, you could find that they were internalizing chunks and vesicles full of tumor-derived material. So to check if this was actually, was following this going to be relevant, we went to the lymph node. We found that we could still find puncta inside these different DC subsets. So these are resident CDC1s, CDC2s, and migratory CDC1s and 2s. And what we found is that we found these puncta, but not only that, they co-localized with other sort of bona fide melanoma antigens, which was great because this was a B16 melanoma with the ZS green. And this suggested to us that if we followed the ZS green, we could use that as a way of following other antigens. So the first thing we did was we tried to understand if it was soluble antigen transport or if it was directly carried in. And so we did this by using CCR7 deficient mice. The tumors grew beautifully. We then looked in the lymph node. And although there were reduced numbers of the migratory DCs, more importantly, we found that we lost all antigen in the lymph node, in the sort of migratory DCs, the resident DCs, even the monocytes, and these were kind of microparticles, so subcellular particles of ZS green. So they all disappeared in the absence of CCR7. We also wanted to address that monocyte question, and we repeated this in CCR2 deficient mice where we lost the monocytes and it had zero impact on the tumor draining of antigen. So we kind of ruled that out and so kind of came to the conclusion that we actually needed cell migration in a CCR7 dependent manner to get antigen transfer. So then to sort of get a rough idea of maybe if it was the migratory DCs, which seemed likely given the CCR7 dependence, we looked at the MFI of these DCs both in the tumor and in the tumor draining lymph node. And we found that as they moved, they reduced the MFI, and we could also see a reduction in the number of puncta containing that ZS green antigen. So this suggested to us that they were going in and possibly handing it off. Obviously, there were other possibilities, but that was just sort of our working hypothesis at the time. So to try and understand that, we developed this assay where what we would do is give our B6 mice these ZS green expressing melanomas. After the tumor had grown, we would purify from the lymph node ZS green expressing, ZS green carrying dendritic cells. And at the same time, we would select non-tumor bearing NTNG mice. So these have red nuclei, different DCs, mix them together, wait overnight, and then start looking for NT positive cells, which now had antigen in them. And what we found from this is, just using this heat map, we can see that the migratory CDC1s and CDC2s were the best sort of donors, and that actually the CDC1s, both the migratory and resident, were the best at sort of acquiring antigen. So what we'd found is that in vitro, we could recapitulate some of this antigen transfer from one cell to another. We then sort of use this a bit more, and we sort of, I'm going to skip over some data because of time, but we sort of showed that we could, if we blocked exocytosis of exosomes, that had no impact, and we did things like that. But interestingly, if we put them over a transwell, we blocked transfer from all of the different donors to all the recipients. And we've got rid of the recipient CD103s because they tended not to survive very well, and so there was just much more variability. So that was great. It looked like they were passing something, and it didn't look like it was exosomes or soluble protein. So we started trying to understand, well, what form was that antigen in? So we actually gave those B16 tumors to those MTMG mice that I spoke about before, and then mixed them with CD45.1 DCs and incubated those overnight. And then we used a lattice light sheet microscope to actually image at very high resolution those recipient cells that had become green, essentially. And what we found is you found these puncta inside that were green, but were surrounded by a red membrane. And then that's shown sort of here in A, but then reconstructed on the right in 3D, where we've taken a slice through that cell, where the purple is the extracellular, the host membrane with CD45.1. Then you can see these MT membranes, and that contains the CS green. So what this suggested to us is that somehow they were transferring antigen, and it was coming over in membrane enclosed vesicles. Since it wouldn't cross a transwell, we thought this might be something to do with cell-cell contacts and some kind of synaptic transfer, which hadn't been described before, but we thought it'd be worth looking for it. So we used these triple reporter mice, which are incredibly beautiful and quite fun to work with, which have XCR1 Venus, CD11C cherry, and CSF1 receptors. So these are MAC blue mice. So you can look for sort of yellow-orange were the CDC1s, red are CDC2s, and blue are the monocytes, and purple could be resident 11Bs. So we just imaged this lymph node live and then looked for interactions. And what we found is that most interactions between cells were very brief, being sort of like less than 30 seconds. But if you looked a little bit further, you found this sort of second set of interactions that were a bit longer. And so when we modeled this mathematically, we found that a bi-exponential decay was the most fit the best, which suggests that we had both short interactions of just cells passing each other, but also more stable interactions where they stuck together for a reasonable amount of time. And when we took the half-life of the second interaction, we found that DCs were forming these kind of 4-5 minute long interactions at times. With monocytes, they really weren't. So it did seem to be a selective thing, sorry, between DCs. So what does that look like? We took advantage of the fact that we had a really amazing lattice light sheet to hand. And so from this one, we purified MT CD103 DCs, so those have the red membrane, and MG CD8 alpha DCs. So these are migratory and resident DCs. And what we did is we put them together on a cover slip, and then we just image them. And this is where we work out if Zoom will like videos. And if it doesn't, I'll just do interpretive dance. But what we found is that the DCs kind of actively probe each other, as you can see from these sort of protuberances, and then they sort of can move together and form kind of mold themselves to each other, which was interesting to us and hopefully to you. So we then moved on and looked at what that interface looked like. And so taking a slice through an interaction, you can see that this synapse is quite active, and there's sort of blebs of membrane forming and moving in between each other. So not only were these interfaces close, but they were also passing material backwards and forwards. And you could look at both cells and see that this was almost that probably was a bidirectional process, which is great, but does it show that we get antigen transfer? Well, we took antigen-loaded CD103s this time, so you're going to see a transmitted light image, but with bright green for the ZS green, and we've put in the NT, so nuclear tomato CD8 DCs. This time we left them overnight, so this is sped up quite significantly. And what we could see is that they form these synapses, and if you look closely at the thing, you can see green starts getting passed across. This has been stabilized on the green cell. It's actually moving around quite actively, so it was still alive and exploring. Eventually, you see that they separate, and interestingly, it then grabs onto another NT positive cell, and you can see that antigen starts getting passed across there as well. So what we can find is that we're getting this antigen transfer from one cell to another. We imaged this multiple times and then worked out how long it took to start getting this transfer. And it was about 4 minutes, 5 minutes. So it looked about similar to what we were seeing in vivo. So we thought that might be a feasible thing. In the interest of time, I'm going to skip past that one, but we then imaged it in vivo and we saw a very similar thing, suggesting that we do have this antigen transfer at synaptic interactions. So does it really mean anything? Because I showed you earlier that when we looked at these DCs, only the CD103s could drive CD8 proliferation in vitro, and only the CDC2s could drive proliferation of OT2s. Well, now that we had a stable antigen that we could track, we could actually control for antigen loading. And what we found is that if we purified only antigen-bearing DCs and then put them with OT1s, we found that actually CDC2s could also drive OT1 response, OT1 proliferation, sorry. And CD8 DCs that are resident in the lymph node could also drive these proliferation. But interesting, they all drove them in slightly different ways. And I'm going to skip very quickly because I've realized the time. But basically, the resident CD8s looked like they were poorer at driving kind of memory responses, which have been shown to be important for immune clearance, tumor clearance. So this kind of relates back to what we were saying about Yanni Borst's idea that maybe these resident DCs normally would do T-cell help, coordinate that, and in their review, they suppose that that might be missing. And this kind of supports that idea that these CD8s, even though they will prime, they don't do the things that we would expect from a robust CD8 priming response. And then just to address cross-dressing, because obviously all the things I've shown you haven't ruled that out. Well, actually, we took some BALB-Cs versus the CD45.1s, sorted the positives versus negatives again, incubated them, and then looked for H2KB transfer. And we found that actually accompanying the ZS green transfer, you'd see this transfer of H2KB. And this is just sort of looking at CD8 alpha DCs, CD103s, CDC2s down here. So we do also get cross-dressing. I can't rule out that that might be important. However, obviously there is sort of cross-dressing and there is presentation of these H2KB on the surface of the resident 11Bs, for example, and those don't stimulate OT2s, OT1s. So it suggests that there's something else going on as well. So now what my lab is working on is really trying to understand why these resident DCs aren't activated in tumor and how they're normally activated, if they are, because actually, you know, a lot of the research on those was done before we really had a full understanding of different subsets. So we're trying to understand that, and also trying to understand the organization of the tumor draining node, because we think that in the absence of sort of robust interferon responses and things like that, you don't get the same reorganization to coordinate some of these responses. So as a result, we've been doing some work on that. And I've realized I've rushed that, but I've also realized there's only 2 minutes for questions. So thank you to everyone at the Beatson who's in my lab and working on stuff like this now. Also, a lot of this was obviously work that I did back at UCSF. So thanks to all the people there. Big thanks to Megan Ruland, who has just started her lab at Oregon and is fantastic. If you have a chance to meet her, you should. And thanks to our funders. Thank you.
Thanks very much, Ed. That was really good. That was really cool. I really enjoyed the videos.
They were a lot of effort.
I can imagine. Well, they look like they were worth it. Fred, did you have a question? I think you had your hand up, or was that by accident?
I was actually looking for the applause emoji and I accidentally raised my hand. But I was hoping we were going to get some interpretive dance there as well.
It would have been so much more disappointing than you're thinking.
No, it's a great story. Thanks very much, Ed.
Thank you.
Yeah, so don't forget, you can ask any questions in the Q&A box or raise your hand. Oh, yep. So Gary has his hand raised. So Gary, you can, yep.
Hi, Ed. Again, yeah, lovely videos. I was just going to ask, what do you think might be the purpose of this in like a normal inflammatory response for just an infection or something?
It's a good question. So I think that my feeling is that this is more to do with things like tolerance, right? So we've got these migratory DCs, which maybe there's sort of a lot of stuff going on in tissues, which could be misinterpreted or whatever. So having that extra layer where you need other signals to come in, activate your lymph node, activate the resident dendritic cells, and those then need to coordinate a second layer of T cell activation. That's kind of where my thinking is. And I guess in my mind, that's where a lot of tumor immunology falls down, in that I think we have that first layer coming through relatively okay, and maybe we change that with some of the therapies, but I think we haven't really thought enough about the lymph node resident DCs and how we might need to improve that to sort of go from just, oh look, we have T cells priming and we have spots in my Ellis spot, through to, oh, we've actually got a robust immune response that's going to do something.
Yeah. Okay, it's interesting. Thanks.
Thanks.
I've got a question from Fawaz.
Hi, thanks. So my question was more functional, and we probably have addressed it in the paper. But does the type of antigen being presented affect this exchange?
So that is a really good question that we have definitely not addressed in the paper, because a lot of what we've done obviously relied on the fact that we could use this very stable fluorescent protein. And when I sort of showed that it co-localized with the melanoma antigens, those are also things that are found in melanocytes. So they're quite resistant to degradation as well. So there is always the possibility that there are other, there's other mechanisms going on as well. But this is just the one that we could study using the system we have. Yeah, definitely interested in the other ones, but we need to work out a way of doing that. That probably takes smarter people than me. I just like microscopes, so it gets a bit problematic when we can't see it.
Thank you.
Thanks.
So we've got a question from Nicolaj, which is, says, thanks for a great talk. I was just wondering, could you clarify a bit on the CCR7 knockout model and how much does it affect ordinary tumor growth mechanisms?
Yeah, so the CCR7 model is thoroughly messed up. Their lymph nodes aren't particularly, they don't develop normally because they've obviously got a lot of things missing. In terms of normal tumor responses, in CCR7 deficient mice, the tumors grow a little bit faster for sure. And you really get no anti-tumor immune responses. So, but then again, that's not surprising because you have none of these migratory DCs coming to the node. But so yeah, that's really all I can say about that model. To be honest, it's one of those ones that we used it as a thing that just because we needed something that did it, but it's definitely not an optimal model for most things.
Cool. And we also have a question from Orhan, who also said, very nice talk. And do you think similar DC interactions occur with NK cells?
Oh, for sure. I mean, NK cells are important for DCs in the tumor. So they're the major source of FLT3 ligand, which is a growth factor, survival factor for CDC1s. They produce a lot of the chemokines, which we suppose lead to tumor ingress, to DC ingress into tumors. So they're definitely important for them. We haven't looked at it in the node. It is something that we're sort of trying to build towards, because when I was saying we're looking at the organization in the node, the thing we've been trying to do is get codecs working, but not codecs where we pay hundreds of thousands for the machine and the reagents, more codecs where we follow the protocol and try and do it on a shoestring at home. And we're getting there. So that is one of the cell types that we've obviously got that we want to understand how that works. And we want to compare it in sort of these robust Th1 responses, for example, influenza, and compare that to these tumor responses where we, to be honest, get pretty weedy T-cell responses. So yeah, but it's definitely a thing we're thinking about.
Cool. Well, that's really interesting. Yeah, so if there are no more questions, then I guess we'll leave it there for today. Ed has put his contact details in the chat box if anyone has any follow-up questions. And yeah, thank you again, Ed and Adriana. That was 2 really great talks. We'll be back the same time next week, where we'll have talks from Fred Sheedy and also from Alexis Mobley. So that should be a really great episode. So yeah, I hope you all have a great week, and see you all next week.
Thanks, everyone.